Hello, everyone, and welcome to today's webinar. Accelerate research with high-throughput friendly gene expression analysis without RNA purification. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, today's webinar is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box at the bottom of your screen and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use this Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I would now like to introduce to you our speakers for today's webinar. Cindy Boardman, Scientist, R&D Sample Prep at Thermo Fisher Scientific, and Luis McGregor, Staff Engineer, Systems Design, Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Presenter tab at the left of your screen. Cindy, Luis, welcome. Cindy, you may now begin your presentation.
Okay, hello everyone. I'm Cindy Boardman, an R&D scientist with Thermo Fisher Scientific. I'm joining you today along with Luis, my colleague, to highlight our automation-friendly gene expression analysis product, Cells to CT Express. So I've got an agenda here. Our agenda today is to start out with an overview of some of the gene expression offerings from Thermo Fisher Scientific, and then move into some finer detail about the specific kit we have selected for this study. And I'll hand things over to Luis, who will discuss the specifics of creating and executing the automated workflow on a liquid handler, after which I'll wrap things up by presenting the data. And we'll take questions at the end, but please feel free to add any questions to the chat as we go along. If we can't get to your question during our session, we'll be sure to follow up with you afterwards. So let's get started. Here we shared, just for your reference, some of the Invitrogen and Applied Biosystems gene expression analysis products from Thermo Fisher Scientific. The one we have selected for our automated workflow is the Cells to CT Express kit, which is highlighted in red. This is a TaqMan probes-based kit for RT-qPCR. With this kit, the researcher must supply only the TaqMan probe-based assay for the genes of interest, and the kit includes everything else needed for cell lysis, DNA digestion, reverse transcription, and qPCR. This is the latest release in the Cells to CT product line, which, for those who may not be familiar, are complete kits for gene expression analysis, which permit reverse transcription and quantitative PCR directly from cell lysates without RNA purification. So, you probably know traditionally the RNA would first need to be purified from the lysates by means such as spin columns or organic extraction before gene expression analysis by RT-qPCR. With the Cells to CT kits, you can just skip that purification in the middle and go directly to RT-qPCR from the lysate. These kits are available in one-step or two-step versions for use with CyberGreen or TaqMan, and also for specialized applications such as single cells or stabilized blood. Previous versions of the kit required the addition of a stop solution. Required the addition of a stop solution. And with this new kit, we've eliminated that step. The unique formulation of the lysis buffer allows you to not have to do that, and the relatively faster throughput and reduction in plastic tip consumption is particularly advantageous for customers performing high throughput gene expression analysis on liquid handlers. Additional features of the kit are the inclusion of a heat labile, double-strand specific DNAs for gDNA removal, and superscript 4 velo reverse transcription master mix for effective inhibitor-tolerant reverse transcription. The workflow for the manual method is shown here. First, a medium is removed from your cells, and the monolayers are washed with PBS. Then the DNAs is combined with the lysis buffer and added to the cells. There's a 5-minute incubation with shaking on an orbital shaker at room temperature. The reverse transcription master mix is plated into the RT plate, and after the 5-minute lysis is up, lysate is transferred into the RT plate. The RT plate is cycled on a thermocycler for a 25-minute cycle, and then the TACMAN gene assay for the gene of interest is combined with the master mix for the qPCR reaction and plated into the qPCR plate. And when the RT is finished, you can transfer your cDNA into the qPCR plate and put it onto the real-time cycler. And now I'm going to hand this over to Luis to talk about the work that he did to automate this workflow.
All right. Thank you, Cindy, for that overview of the CellSaTT Express kit. Once again, my name is Luis McGregor. I'm a staff engineer here at Thermo Fisher Scientific. I'm going to be going over the automated workflow that was designed for the CellSaTT Express kit. So I'd like to start with just talking about the system overview. We chose to use the TCAN Fluent 780, which is an automated liquid handler. This system has 2 automated pipetting arms, one of which is a flexible channel arm, which is an 8-channel arm capable of extending and contracting its channels to be compatible with different plate formats. It's also capable of utilizing individual channels or using all 8 channels all at once. The second pipetting arm we have is a multi-channel arm. This is a 96-channel arm capable of pipetting liquid with 96 tips simultaneously. We chose the TCAN Fluent 780 because that was an automated liquid handler that we had on site. But I just wanted to express that this automated workflow is designed to be compatible with all other common liquid handlers on the market. I just put a few here at the bottom that obviously we're using TCAN Fluent 780. There is a smaller version, which is the 480. Also, this is going to be compatible with a Hamilton Star, Starlet, or a Beckman Coulter Biomech i5 or i7 series. So what we have here is the work table that was designed to accommodate for this entire automated workflow. Just starting from 1, we have this is our deck configuration picture at the bottom left, and then a 3D rendering on the top right. So if you look at the bottom left, starting with number 1, this is room for our 200 microliter tips. These are tips going to be utilized for the 8-channel arm. For 2 and 3, these are tips utilized for the 96-channel arm. We have our 50 and 150 microliter tips. Going into our reagents, number 4, we have a 300 ml trough that's going to be utilized to house our PBS wash. We have a 96 depot plate that we're utilizing as a reservoir for our express lysis solution. And we also have another 300 ml trough that's going to be empty but utilized as a waste trough for the process. 7, 8, 9 are going to be our plates that we're going to be using for this process. Number 7 is our 96 well semi-skirted plate. This will be our RT plate, our reverse transcription plate. We have number 8, which is our 384 well real-time PCR plate. This will be utilized for qPCR. And then 9, we have our 96 well flat bottom plate. This will be our input plate that has our cells adhered to it. This will also be utilized for cell lysis. And lastly, we have our 5-position tube holder, which is going to hold 2 ml flip cap tubes. This will contain our RT and qPCR reagents. So this is just an overview of the entire process, the automated workflow. This workflow consists of 3 processes that I'll be going to each of them in detail. They're signified right there with that robotic arm, process 1, 2, and 3. Process 1 is going to be specifically cell wash, lysis, and RT prep. The blocks that are in blue are going to be on the actual automated liquid handler. The blocks that are in gray, those are going to be off deck. And so we'll actually put that onto a thermocycler to perform reverse transcription. After that's complete, we can move back to the liquid handler for process 2, which is going to be qPCR prep, specifically dispensing our qPCR reagents into our plate. Next, we'll do the second part of qPCR, which is going to be dispensing our sample into our qPCR plate. And at that point, we'll be able to take the plate off deck and then perform qPCR. So this is, we're going to be starting with process 1. Again, this is going to be cell wash, lysis, and RT prep. The objective for this process is to utilize our system to wash the cells and prepare lysate for reverse transcription. So on the left here, I have pre-automation. This will just be prior to putting in the plate onto the automated liquid handler. We would have our cells adhered to the plate. We would then put that plate onto the automated liquid handler, and the first step there would be to remove our culture media out of that plate. We would then wash our cells with PBS, add our lysis solution, and then there'd be 2 steps that would be being performed simultaneously. One, we have an on-deck orbital shaker that will be rotating and performing that lysis step. At the same time, the system will then be dispensing our RT reagents into our 96 PCR plate. After the lysis is complete, we will be transferring our lysate from the culture plate to our 96 PCR plate. And at that point, we would then take the plate off the liquid handler and then perform reverse transcription. So this is the work table once again, but specifically the items highlighted in red are going to be utilized for this first process. Once you start with 1, we have our 200 microliter tips for our 8-channel arm, our 50 and 150 microliter tip boxes for our 96 channel arm. We'll again be using our 300 ml trough for PBS, 96 depot plate for express lysis solution, and then a blank or empty 300 ml trough going to be utilized for our reagent waste. And then we'll also be using we'll have a 96 semi-skirted PCR plate on deck that'll be utilized for our reverse transcription. And then we have our 96 flat bottom plate, which will be our input plate with our cultured cells, and also used for lysis plate, as a lysis plate. And then we'll have 3 2-mil flip cap tubes that'll be utilized to house or hold our RT cocktail reagents. So prior to the automation, there are 2 reagents that need to be prepared prior to starting process 1. One of which is our express lysis solution, which is a combination of our express lysis solution and our Invitrogen Express ezDNase. These volumes for both of these reagents are based directly from our user guide. And you'll see a little bit of finer print at the bottom here. What this is you may have noticed that we're utilizing a 96 depot plate as our reservoir for our express lysis solution. The reason for that is to try to, every reagent reservoir, when it comes to automation or manual pipetting, will have a amount of dead volume or volume that cannot be pipetted out of the well at the end of the pipetting process. And so to make sure that we accommodate for that volume in there, the 96 depot plate that we're using for lysis has a dead volume of about 30 microliters per well. So to accommodate for that, we are adding an additional 2.88 mils of this lysis solution, which is 30 microliters per well for our 96 well plate, into we'll add that 2.88 total to 5.28 as our total to accommodate for that dead volume and ensure we have enough volume for pipetting. So that's the lysis solution, and we're also going to create our RT cocktail, which is our superscript for Velo master mix, as well as nuclease-free water. And so this will actually be in a 2-mil tube, which has a small dead volume on it, which is about 20 microliters per tube. So we're utilizing 3 tubes, so we're going to be adding an additional 60 microliters to that 4.23 to accommodate for that dead volume and ensure sufficient volume there as well. So going into process 1, starting with this first 3 steps are going to be removing our culture media and washing our cells. So all 3 of these steps will utilize our 96 channel pipetting arm. What we're going to do first is going to be aspirating off 160 microliters of our culture media out of the culture plate and discarding that into our waste trough. We will then be utilizing the same arm to pipette in 100 microliters of PBS into that culture plate as well. And then after that, we'll be removing that PBS wash, about 120 microliters of PBS that will be pipetted off and dispensed into the waste trough. And so I added a little asterisk here because with this kit, it is recommended to remove as much PBS as possible prior to cell lysis. And so a couple of things that we did and some recommendations I have on this is trying to aspirate as low to the bottom of the plate as possible in order to pull up as much media as much PBS as we can. I also increased the aspiration to 100 and by 20 to 120 microliters in order to pull up as much as possible, so we can reduce the amount of dead volume or residual PBS in the well after aspiration. And during our automation process, we actually had about 15 microliters of residual PBS left in that culture plate, and that still yielded acceptable results. So the second part of process one is going to be specifically about preparing our lysate and distributing our RT reagents. So this actually the first step will utilize the 96 channel arm, and the third step here will actually utilize our 8 channel arm. So first we're going to be adding our express lysis solution to the cells. This would be 50 microliters that'll be dispensed from the reagent trough into the culture plate. At that point, the culture plate is sitting on top of our orbital shaker. So the culture plate is then shaken on the orbital shaker to complete cell lysis. And just the parameters we have for our shaker are at the bottom here. We had an acceleration of a 5-second ramping time getting to our ideal RPM, which is 900. And we had that running for about 5 minutes. And as for the last step, or the third step here, we would be then utilizing our 8 channel arm to add our previously prepared RT cocktail, 40 microliters of that from our 2 ml tube, and dispensing that into our 96 well PCR plate. And just a recommendation here is this step of adding the RT cocktail to the PCR plate can actually be performed at the same time during that 5 minutes that the plate is shaking on the orbital shaker, and just to maximize time there and make sure it reduces time a little bit there. And then the last few steps on process one is then going to be transferring our lysate. So we're going to utilize the 96 head this is when the 5-minute lysis is complete, we will then utilize our 96 head to pipette 10 microliters of the lysate from the culture plate and dispense it into the 96 well PCR plate, which has our reagents already in it. And after the dispense has been made, we add a mix step there to mix our lysate and our RT reagents, utilizing the same tips, and really just to homogenize those as much as possible before reverse transcription. Our mix parameters that we used are 40 microliters, 4 cycles for mixing. And at this point, our automated process one is complete. The 96 well PCR plate is ready for reverse transcription on a thermocycler. We'll perform that reverse transcription. It's about 25-minute cycling time. And while this is being run, we can actually continue on to the process two while the reverse transcription is being performed. So now we're into process two, which is qPCR prep, specifically reagent distribution. So the objective here is to utilize the system to distribute our qPCR reagents to our 384 well PCR plate. So in the design of our experiment, we actually analyzed the expression of 2 genes. So what the automated liquid handler would be doing is dispensing the master mix for gene 1 into 384 well plate, and then we'll continue to do the same thing, but we'll dispense our master mix for gene 2 into the 384 well plate. So for process 2, once again, this is our deck configuration. Highlighted are the components that we need for process 2. We'll be once again be using the 200 microliter tips, the 384 well PCR plate, which is on number 2, and that'll be for our qPCR. And then we'll also utilize 2 2-ml flip cap tubes that are going to contain our qPCR cocktail. So similar to process 1, there are a couple of reagents that need to be prepared prior to starting the automation. We're going to make 2 qPCR cocktails. Again, in this particular design, we are analyzing the expression of 2 genes. So our first is going to be CDK4 gene. We're going to be adding our fast advanced master mix, as well as our gene expression assay and nuclease-free water. And then for our beta-actin gene, which is our second gene, we're going to be adding the similar reagents there. And we're going to be preparing that for 192 reactions. We are putting this into a 384 well plate. So we will split the plate wells in half, 192 for one gene and 192 for the second. And once again, taking dead volume into consideration, we again are going to be using 2 ml tubes, which have a dead volume of about 20 microliters. So 20 microliters will be added to that 1.54 mils for each of these cocktails to accommodate for that dead volume and ensure there's a sufficient amount of volume prior to pipetting. So for process 2, we're going to be specifically distributing our qPCR reagents. Both of these steps are going to be utilizing our 8-channel arm. What we'll be doing is we're going to be adding 8 microliters of qPCR cocktail for gene 1 or CDK4. We'll be aspirating that from our 2 ml reagent tube and dispensing it into our 384 well PCR plate. We'll be distributing those reagents to quadrants 1 and 2. Once again, that'll fulfill our 192 samples in the plate. That'll be half the 384 well plate. Once that's complete, we'll utilize the same arm to do a very similar process, 8 microliters from the qPCR cocktail for gene 2, which is our beta-actin gene, will be dispensed from its own 2 ml reagent tube that was prepared prior to automation and dispensed into the 384 well PCR plate into quadrants 3 and 4, are the remaining 192 wells of that plate. And at that point, our automated process 2 is complete. Our 384 well plate is ready for cDNA addition after reverse transcription has completed. So going into process 3, this is going to be our sample distribution for qPCR prep. Process objective here is going to be utilizing the system to distribute our cDNA or our RT reaction to our 384 PCR plate. Process 3 cannot be started until the reverse transcription has completed. Once that's completed, that plate can then be placed onto the automated liquid handler. That's when process 3 would start. It would transfer our cDNA to our 384 well plate. And after that, we then take that plate off and then run it on a Quant Studio or real-time instrument to perform qPCR. So for process 3, this is our work table configuration. We need 3 components. We need our 50 microliter tips, 4 boxes of those for our 96-channel arm. We'll also be utilizing our 96 well semi-skirted PCR plate. At this point, this contains our cDNA because this is post reverse transcription. And then we'll also be using our 384 well real-time PCR plate, which currently contains our qPCR reagents already in it. That was completed in process 2. So for process 3, for sample distribution, we'll be doing just that. We'll be distributing our cDNA. So we'll be utilizing our 96-channel head here on this first step. And we'll be dispensing 2 microliters of cDNA from our 96 well PCR plate to our 384 well PCR plate. And after each dispense, we will be performing a mix to homogenize the qPCR reagents that are already in that plate, as well as our cDNA. And this is going to be 384 wells. And so we'll actually perform 4 transfers from the reverse transcription plate to the 384 well to dispense 2 microliters into each well of that plate. The mix that we're performing after each dispense is 4 microliters at 4 cycles. And at this point, our automated process 3 is complete. The 384 well PCR plate is ready for qPCR. And that qPCR has a cycling time of about 40 minutes. So that concludes the overview of our automated workflow. I'll then pass it back to Cindy now for her to discuss some of the data we collected and comparing that to the automation data as well as the manual data.
Cindy, I'm so sorry. It looks like you are muted. Your AirPods might be muted. No problem. Just Nope, not yet. No problem. It sometimes happens with the AirPods. Do a quick test. Nope. Okay, no problem. We'll get you back. You can always, there we go. I think we got you back. Yep, here we go. You're all set. Go ahead.
Awesome.
I think we got you back. I can hear you.
I don't know.
Yes, I can hear you.
Oh, is it sounds good now?
Yes.
Okay, I guess you can hear me. I can't. Okay, sorry about that. A little technical difficulties. And let's get to the data then. What we've done for this study is to seed 5,000 mammalian cells into 96 well plates. And then after an overnight incubation, we processed one plate by the manual method and the other plate with the automated method. And so here we've shown some amplification traces of those two different plates. We chose to prepare half of each qPCR plate with beta-actin, TACMan gene assay, and the other half with cyclin-dependent kinase 4. And we prepared duplicate reactions for each RT reaction for each gene. So these are the plots, and you've got the beta-actin in red and the CDK4 in blue. And we have our manual method on the left and our automated method on the right. And here we see the actual CT mean values obtained for the 192 replicates for each gene using the manual in red versus the automated in gray workflows. The error bars represent one standard deviation. And so you can see that we've obtained comparable results between the two methods and without having to purify RNA. So the manual workflow makes sense if you have just a few plates, but for larger studies, the ability to automate the CELSA-CT workflow for higher throughput is a tremendous benefit. And for your reference here, I've just put the link to the application note if you want to look that up and see more details of how we did this. But, to conclude, CELSA-CT expresses a fast, sensitive, and automatable solution for high-throughput gene expression analysis. The revised and upgraded components improve the performance relative to earlier versions of CELSA-CT, and the removal of the stop solution from the workflow helps to save time and pipette tips. Luis has successfully developed a straightforward automation workflow for the kit that does not require custom labware or carriers and is applicable to most liquid handlers on the market and produces results that are comparable to those produced by the manual workflow. So thank you very much for your attention, and we can get to the questions now.
Cindy, Luis, thank you so much for that excellent presentation. We're going to bring Luis back up here as well. Okay, so yes, it is now time for our live Q&A portion of our webinar. And to our audience, if you haven't already done so, please submit your questions now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. Okay, so we've got some great questions coming in. I'm going to dive right in. Luis, First question, could you process the first step in 384 wells? Seed the cells in 384 wells, wash and lyse them?
Yeah, I think from my engineering standpoint, you could technically use a 384 well until 96, but you would have to, in the process for 96 well, we are utilizing, I think, at least 160 microliters of our culture media. So obviously that'd be significantly less because that's not possible in a 384 well plate. But if you are able to scale down all of those reagents required and your media itself, you could technically do that process up front, and that lysis then would be performed on that orbital shaker as well. You would just have to have probably a different plate adapter to hold that 384 well plate.
Great. And Luis, I'm going to go with you for this next question as well. Is the automation of the cells to CT Express kit limited to a throughput of 96 samples?
That's a good question. So in the automation workflow that we designed, we did 96 samples as an input, but really this automated workflow is designed to be scalable. So really your only limiting factor is having enough space on your automated deck to hold all the plates necessary for the workflow. So you could do that in two ways. You could either have a smaller system that has plate hotels or plate carousels attached to it to house those extra plates, or you could just utilize a larger automated liquid handling system that could have all those plates on the deck. So really it's as big as you have space to put the plates on, as high as you can scale this automated workflow.
Thanks, Luis. So Cindy, I'm going to pop over to you for this next question. Is the cells to CT Express lysate inhibitory to downstream RT-PCR? Oh, Cindy, now I don't have your audio. You know what? Technology is great until it isn't sometimes. Let's get you set again. Give me a test here. Not yet. Cindy, real quick. Nope. Luis, I'm going to pop over to you and have you answer a question while we troubleshoot with Cindy. Luis, is it possible for this automated workflow to be a full walk-away solution?
Yes, it is. The workflow that we went through today does have two off-deck steps that are completed. The reverse transcription and qPCR completion are all done off-deck, but they do have on the market, there are thermocyclers that are integratable onto your automated liquid handlers that could perform those steps. So if you had that on there and had enough space on your deck on your liquid handler, you could perform those two steps. Those two steps, including the three processes that I went over, all in a walk-away solution.
Very good. And Cindy, I have some questions for you. Let's test your mic real quick. You may not have audio here. Okay, Luis, I'm going to bump to another question.
Okay.
Is this automated workflow only applicable to the cells to CT Express kit?
No, it actually is. We only went through the cells to CT Express kit, but this could be applied. This workflow could be applied to other kits. As Cindy talked about earlier, specifically the cells to CT Express kit is our most recently released one and does not need the stop solution. If you were to incorporate another previously released cells to CT kit for automation, you could utilize the same workflow that I talked about earlier today. The only difference was you would need to add an extra pipetting step to add that stop solution into your plate, and there would be an additional 2-minute shaking prior to reverse transcription. But other than that, the workflow that I went through today could be applicable to other kits that we have for cells to CT.
Excellent. Sounds good. Cindy, let's give you a test. Still working on that audio. Luis, I'm going to keep going with you here. I don't have an orbital shaker on my liquid handler. Is there an alternative?
So, I think the orbital shaker would be the best, I guess, component to perform that, shaking on the lysis. But if you don't have one of those, you could utilize that 96 channel head to actually slowly pipette up your liquid up and down in a uniform fashion to make sure that mixes well enough. So that would be a good alternative if you don't have an orbital shaker. It's just pipette mixing.
Very good. Thank you, Louise. And sorry to be picking on you here. Cindy, how are we doing over there? Cindy, can you hear me? I can't hear you yet, Cindy. Well, let's see. I think a lot of these questions are for Cindy. Let's see. Cindy's microphone is not working. Let's see. Louise, help me out. Is there any questions on here that I can address over to you? Let's see. I will say we do have a question coming in asking about on-demand viewing of this presentation. Yes, today's presentation will be available for unlimited on-demand viewing. Once today's live presentation is complete, it will process and Laborats will send you an email letting you know when that on-demand viewing is available for replay, and you can watch it unlimited. We will send you that email, and we do encourage you to send that email to any colleagues and friends of yours that may be interested in today's webinar. Okay, I'm going to have you try to take some of Cindy's questions here. I'm going to ask this one again. Is the cell to CT express lysate inhibitory to downstream RT-PCR?
So, well, the kit has been developed to have excellent performance. And when used as directed, results are comparable or even superior to those from purified RNA.
Excellent. And Cindy, I'm going to test your mic again. I know you can hear me, but darn it. Shoot. Okay, let's go with this one. Louise, you're in the hot seat. How does cell to CT express compare to purified RNA?
We have tested the results on many cell lines and found that compared side by side, the cells to CT express produces a linear response over 5 logs of cell input and is often more sensitive than purified RNA.
Excellent. And if there's any questions that come in from our audience that we are unable to answer live today, we will answer those questions via the email address you provided at the time of registration. So let's see, Louise, I'm going to pick on you again. Here we go. Is DNA contamination a concern with the cell to CT express?
Well, while we do recommend using intron spanning primer sets whenever possible, the inclusion of ezDNA effectively removes genomic DNA during the lysis step. So,
excellent. And you may know the answer to this one. Can I use cells to CT express on any cell type?
I think we have evaluated cells to CT performance on 13 commonly used cell lines and obtained results as good or better than those obtained with purified RNA.
Great. And speaking of purified RNA, how does the cell to CT express compare to purified RNA?
We have tested the results of many cell lines and found that comparable side to side. Well, I'm sorry, can you repeat the question?
I absolutely can. How does the cells to CT express compare to purified RNA?
Okay, I think I did hear. Okay. Yeah, I was just saying we have tested the results of many cell lines and found that comparable side to side. Cells of CT Express produces a linear response over 5 logs of cell input and is often more sensitive than purified RNA.
Excellent. Thank you. And I'm going to wrap with this final question. Luis, how does cell to CT Express compare with other direct lysis kits?
That's a good question. A side-by-side comparison with direct lysis kits from other manufacturers has shown that cells to CT Express produce the lowest CT values. Differences in CT values are represented in some cases many-fold differences in detection.
Excellent. Thanks, Luis. And I know we gave you a lot of questions today, but before we close, do you have any final comments for our audience?
No, I just want to thank our audience. Thank you very much for your time. And once again, the workflow that we went over today is summarized in an application note that we made for the Cells to CT Express kit. I believe that's attached as a resource on the registration page for this webinar. But it also can be found on our Thermo Fisher website underneath our Cells to CT Express kit.
Thank you so much, Luis. And thank you to Cindy as well. We apologize for the technical difficulties. Sometimes that does happen. And, unfortunately that did happen today. There were quite a few questions we were unable to answer. We will get those questions answered to you via the email address that you provided at the time of registration. And additionally, you may submit questions during the on-demand period as well, and we'll get those answered to you via email. Now, before we go, I do want to thank our audience for joining us today. I also want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. As I mentioned before, today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Until next time, everyone, thanks for joining us. Have a great day, and we hope to see you soon. Bye-bye.